So welcome everyone. The topic of my PhD is the role of gut microbiota in cystic fibrosis. My vision is to offer a complication-free life for people with cystic fibrosis by investigating the role of gut microbiota in the disease. Here you can see the list of my projects. In both of my projects, I try to investigate the composition of gut microbiota in cystic fibrosis. So my first project is the comparison of gut microbiota in cystic fibrosis patients and healthy individuals. So a few words about the background. Between 70,000 and 160,000 people are living with cystic fibrosis worldwide. You can see the gut wall of a healthy individual and a cystic fibrosis patient here. While the CFTR protein in an otherwise healthy individual works just fine, it is dysfunctional in a cystic fibrosis patient. It results in several systemic and local complications, including a continuous inflammatory cycle. These all contribute to a potential alteration of the gut microbiota, so our aim was to compare the gut microbiota of CF patients and healthy individuals. We included both adults and children. Our outcomes were alpha diversity, beta diversity, and the abundance of certain bacteria. In our systemic search, we searched 5 databases with the following search key that you can see at the bottom of the slide. After the selection of more than 2,500 articles, we ended up with 12 articles and 2 additional ones from citation chasing. We finished analyzing alpha diversity and relative abundance. However, the analysis for beta diversity is still ongoing. This is the first result that I'm going to show you. It is for Shannon index, which is an alpha diversity measure. It takes bacterial richness and evenness into consideration as well. It ranges from 0 to infinity, with the higher numbers indicating a higher diversity. The normal population is thought to have a Shannon index between 2 and 4. We compare the standardized mean differences between CF patients and controls. We could form 3 subgroups, infants, children, and adults. And if the articles lie on the left side of the line of no effect, it means that CF patients have a decreased diversity compared to controls. If they lie on the right side, it means it is increased. So in the first subgroup, infants, there was no difference between the 2 groups in terms of diversity. Then in the subgroup of children, alpha diversity was significantly decreased in CF patients. So we can conclude from this slide that there is a significantly decreased alpha diversity in children with CF compared to controls. This second slide shows us the results for observed O2s, which is also an alpha diversity measure. It only takes bacterial richness into consideration. It ranges from 0 to infinity, with the higher numbers again indicating higher diversity. We had 2 subgroups, children and adults. We can see that in the subgroup of adults, CF patients have a decreased alpha diversity compared to controls. So we can conclude that alpha diversity measured by observed O2s is decreased in CF patients compared to controls. On this slide, you can see the longitudinal analysis of Shannon index in infants with CF and controls throughout the first year of life. So the first time point when their stool was analyzed was 4 months, then 6 months of life, and then 12 months of life. If you look at the diamonds, you can see a decreasing trend of alpha diversity in infants with CF compared to controls. This aggregated forest plot shows our results for relative abundance. Relative abundance is a percentage measure. It tells us the percentage of a certain bacterial taxon compared to all the bacterial taxa found in that given sample. We also measured standardized mean differences between the 2 groups. The first column shows the name of the bacterial taxon, then the taxonomic level, then the number of CF patients, and the number of controls. If the point estimate lies on the left side of the line of no effect, it means that that given taxon is decreased in CF. If it lies on the right side, it means that it's increased. You can see that there are alterations between CF and control patients. However, if you look at the bottom of the slide, you can see that Veillonella and Escherichia coli are significantly increased in CF patients compared to controls. So we can conclude that there is an altered community structure in people with CF, marked by a significant increase in abundance of Veillonella and Escherichia coli. We have some strengths and limitations. Our strengths are that we could analyze age-related differences, and we could also analyze the gut microbiota maturation in the first year of life. And we have a higher level of evidence due to the larger overall sample size. However, we have the high risk of bias of the articles, and only high-income countries were represented in our analysis. So in conclusion, I can say that CF is a barrier to the maturation and diversification of gut microbiota. As for implications for research, more articles are needed investigating therapeutic microbiota modulation, as well as CF-related factors influence on gut microbiota composition. So we are almost done with the analysis. Here I wanted to show you the list of conferences that we have or will present our results. Here's a picture of us. In our second project, we will try to investigate the effect of modulator therapy on gut microbiota in CF patients, which is a registry analysis. So about the background, CF comes with several gastrointestinal manifestations, including dysbiosis. The first core problem in the development of dysbiosis is the CFTR gene mutation, which results in insufficient CFTR protein level or function. We have a solution for it so far, the CFTR modulator therapy, which acts by either improving the processing and delivery of CFTR protein to the cell surface or enhancing the function of the inactive ones. The second core problem are all the other factors: exocrine pancreatic insufficiency, low intestinal pH, constant medication, including antibiotics, high-fat diet. So with both these problems contributing to the development of dysbiosis, our question really is, to what extent is modulator therapy effective in resolving dysbiosis? So this was the question we aim to answer. We include children with CF. Our exposure is modulator therapy. The control is no modulator therapy. And our outcome will be the composition of gut microbiota in different time points. Our hypothesis is that dysbiosis is reduced after giving modulator therapy to these children. So our data source will be the cystic fibrosis-related pancreatic disorder registry at Heimpal National Pediatric Institute. We have finished database filtering and data cleaning. The next step this month is microbiota analysis, followed by biostatistical analysis of the results. We will analyze 25 patients at 4 time points at baseline, 6, 12, and 24 months after giving modulator therapy. So once again, here you can see my project list, and thank you for your attention with the quote, "The man who moves a mountain begins by carrying away small stones." Thank you.
Congratulations, it's a great project. It was really good to see that your second project built on the first one and not just loosely related to each other. So it's a big strength of your work.
Thank you.
I had many questions related to this project. We see many patients with cystic fibrosis where I work, and I had 2 main questions or suggestions. First, so the compliance of the patient matters a lot, especially when they get teenagers. So sometimes if they move along with the treatment protocol and they keep the medication, they take the medication as they recommended, it changed the outcome a lot. That's what we see, at least. And also a question maybe that the other interesting thing in this project is that in some patients the gastrointestinal manifestation present first and then the pulmonary manifestation, and in other cases it's opposite, and maybe it would be a meaningful question for the future to investigate why this happens, and as far as I know, there is no explanation for that, right now.
Yeah, I do not know any explanation for it either.
Yeah. All right. So, any question from the audience?
I would have one.
Okay.
So, congratulations to nice presentation.
Thank you.
I would like to, because microbiota is influenced by many co-founders like obesity, socioeconomic, whatever. So I would like to ask about your control in your first project. How they ensure that the control match the intervention, the exposure group?
It's a very good question, thank you. So we only included studies that included control patients with no disease or drugs influencing gut microbiota. So we try to make sure that no one's gut microbiota in the control group was affected by these factors. We also did not include any CF groups that took modulator therapy, of course, because it also potentially influences gut microbiota. So that's how we try to make sure that none of these factors were there.
And if I may, I would have a second question.
Of course.
Second one would be if you are expecting the benefit from reducing the dysbiosis, would you consider fecal microbiota transplantation in this patient? And as far as you know, do these patients have some benefit from this approach?
Yeah, well, as far as I know, the guidelines only extend to diarrhea in terms of, and Clostridium difficile in terms of fecal microbiota transplantation. So there's no current guideline for cystic fibrosis and the related gastrointestinal complications. We also do not know who's got microbial, I don't know, sample or we would give to these patients what is the best for them. So that's also a matter that needs to be investigated. So I do not have any final answer for this question, but it is a good point and it needs to be investigated, I think.
Thank you.
Thank you.
Right, I have one question regarding the first plot you presented.
Right.
In the infants, I remember there was no difference between the intervention and the control group. But the older they get, the less the diversity in the CF group. So what kind of protective factor you can think of in the infant group?
So if we look at this slide, we can see that the diversity switches up. I'm sorry, after the age of 6 months, which is the age around when they start other feeding other than breastfeeding. So it can have a potential effect on gut microbiota as well as closer contact with their mothers at an earlier age. Also, the mode of birth can affect gut microbiota. So if it's a C-section or vaginal birth, it also matters. Yeah.
And where are these data available in the articles, like the delivery, for example?
Yeah, it was, but not in terms of outcomes. So they reported how many of the CF patients were breastfed, how many were formula fed, but when they reported the outcomes, they did not report it separately. Yeah.
All right. And just one last question. Why have you, or why do you have SMDs? What is the methods to calculate or to investigate the alpha diversity different among the studies, or what's the reason?
When you calculate Shannon index, it matters greatly how many bacterial taxa you have found in that sample. And as this number differs among samples, this measure needed to be standardized. So it could influence Shannon index in all patients.
Right, but the same method was applied in all the studies?
No, so in these studies, Obserdotus was applied, but of course we pulled together studies where the same method was applied.